In a groundbreaking discovery, researchers have found that decision-making is not confined to specific regions of the brain, but instead involves a widespread network of neurons across the entire brain. An international team of scientists, part of the International Brain Laboratory, IBL, has created a comprehensive mouse dataset that challenges the conventional understanding of decision-making processes. Anne Churchland, a neurobiology professor at the University of California, Los Angeles, and a founding member of the IBL, explains that numerous areas of the brain are engaged during even seemingly simple decisions. The IBL was established in 2017 to address the limitations of small-scale studies that focused on only one or two brain regions at a time. By combining the efforts of multiple labs, the IBL aimed to explore how the brain integrates information and makes decisions on a larger scale. After years of standardizing methods and instrumentation across the 12 participating labs, the IBL team constructed a brain-wide map of neural activity in mice as they completed a decision-making task. The resulting dataset, published in Nature, reveals that the activity associated with choices and motor actions is widespread across the brain. A companion paper also published in Nature shows that the same holds true for decisions based on prior knowledge. The IBL labs generated the map by pooling neuropixels recordings from 139 mice that completed a visual discrimination task. Each lab recorded different brain areas, covering the entire brain. The dataset spans almost 300 brain areas and includes activity from over 620,000 neurons, making it one of the richest datasets on decision-making. Floris de Lange, a professor of brain, cognition, and behavior at Radboud University, notes that the IBL has made the dataset public, enabling researchers to test new ideas in a powerful and accessible way. The IBL chose a visual behavioral task that was complex enough to capture various aspects of decision-making, but simple enough to be replicated across labs. Mice were presented with a black and white striped circle on either side of a screen and had to maneuver a tiny wheel to center the circle. Upon success, they received a sip of water. Failure resulted in white noise. Electrodes recorded brain activity as mice observed the visual cue, made their choice, moved the wheel, and received feedback. As soon as the animals saw the visual cue, activity appeared in expected areas, including the visual cortex, thalamus, and prefrontal cortex. However, choice-related activity was also present in subcortical areas such as the hindbrain and cerebellum, challenging the assumption that only certain regions encode decision-making information. Movement and feedback-related signals were similarly widespread, with 81% of recorded brain regions containing information that could predict the animal's wheel speed. Activity from nearly all recorded brain regions accurately predicted whether the mouse had received a reward or not, with stronger activity in the thalamus, midbrain, and hindbrain. The findings suggest that the current model of decision-making and the brain regions that control it may be limited in scope, and other unexplored areas might play a crucial role. Although the analysis shows that a distributed network of brain regions contains information about decision-making, even at early stages of sensory processing, future studies need to determine how that information is used for decision-making and learning. The comprehensive map sets the stage for further experiments and could serve as a valuable resource for neuroscientists to validate results in their own labs. These studies emphasize the importance of large-scale multi-lab efforts, particularly for studying brain activity. The global consortium has since expanded to include 21 experimental and theoretical neuroscience labs and has established a new group called IBL 2.0. They plan to share the tools and expertise gained with new partners, and Churchland hopes that their work will inspire larger collaborative efforts, leading to critical insights for the field.